Hello friends, this is Manpreet Kaur with student ID 3011603007. Here I come to deliver my presentation of food science project on the chosen research article titled Coliform Bacteria for Bioremediation of Waste Hydrocarbons. As we know that environmental pollution with spilled oil and waste hydrocarbons has become a problem of major concern on the global scale, as these compounds contaminate water and terrestrial as well as atmospheric ecosystems. These days, the value is constantly increasing because of the ever-increasing human activities related to oil extraction from reservoirs, transportation, and using them as energy source. In order to remove the spilled oil, some physiochemical or microbiological mediums can be used. Physiochemical processes are not considered generally environmentally safe, as the utilization of sorbents, dispersants, and gelators can cause additional pollutions. However, an alternative and effective approach to the solution of the problem is associated with the bioremediation means, as this method is more economical and eco-friendly as compared to the physiochemical one. For bioremediation, two practices are generally suggested. One is biostimulation and another is bioaugmentation. For the bioaugmentation, the oil contaminated areas are inoculated with cocktails of hydrocarbonoclastic microorganisms. Hydrocarbonoclastic microorganisms are the hydrogen degrading bacteria which utilize hydrocarbons as the source of their growth. For biostimulation, it depends on enhancing already existing microorganisms by adding nutrients, surfactants, and other materials. For this article, certain methodologies were followed, which will be discussed one by one. But before going ahead, I would like to throw a brief light on the coliform bacteria. Coliform bacteria are the facultative anaerobic bacteria which generally inhabits the digestive tract of warm-blooded animals including humans. Therefore, E. coli it forms the 10% of the domestic sewage microflora of the worldwide. And in this study, for the first procedure, which is counting and characterization of coliforms, 3 samples from local raw domestic sewage were collected and were used to enumerate and isolate the indigenous coliforms. Indigenous coliforms were then counted and differentiated as per their characteristic morphologies, where Enterobacter colonies were pink with dark centers, Klebsiella colonies turned out to be pink, and E. coli colonies were black with green metallic sheen. These isolates were further processed for their potential for acid and gas production from lactose fermentation at 37 degrees Celsius and 44 degrees Celsius. And the results were as shown. All the isolates showed positive for acid production and gas production at 37 and 44 degrees Celsius, and the total count were calculated as 9.5 into 10 raised to the power 5 and 9.8 and 9.6 into 10 raised to the power 5 CFU per ml. Another methodology which was used for the experiment is hydrocarbon utilization potential for coliforms. All the coliform strains were tested for their ability to grow on a mineral medium with oil vapor as the sole source of carbon and energy. Also, to study the range of pure hydrocarbons that could be utilized by individual strains, a common inoculum strategy was applied. And the cultures were incubated at 30 degrees for 15 days. And as per the results, all the isolates they tested quite good for the hydrogen consumption, and the range it varied from 15 to 30%, and for E. coli specifically from domestic sewage, the percentage consumption turned out to be 22.2 plus minus with a 3.2 ratio. Third was hydrogen uptake by coliforms. The crude oil samples, the crude oil samples were then examined for the two strains of E. coli. One was isolated from domestic sewage and another was from wheat straw. This was done using transmission electron microscopy, and for this, the biomass samples were fixed with glutaraldehyde in the buffer solutions. The samples were embedded in Epon resin, and the final products and the final sections were then mounted on carbon-coated grids and were viewed under electron microscope. Here is the result of the TEM images, which showed the cell cytoplasm of hydrocarbon incubated cells in contrast to that of cell harvested from nutrient broth, which showed enrichment with less electron dense areas with varying dimensions. Another procedure was of oil removal in sewage batches. For this, aliquots of 100 ml freshly harvested raw sewage were taken into 50 ml flask with separation of 1.0 and 5.0 gram of crude oil. All other ingredients were kept same, the proportions were kept same, and the flask were then sealed to avoid hydrocarbon volatilization, and the cultures were then incubated at 30° for 20 days. And the results of these can be seen in the form of the graphs. For the first one, which implies for 3% crude oil, the colony forming units of coliforms, they tend to have they tend to show a spike. Here in this, the purple line employs for total CFU, which goes from 1 to 26.5 for the 3% crude oil. For 1% crude oil, it turns out to be 52.7. For C18, it turns out to be 18.9, while for phenanthrene, it is 15.8 into 10 raised to power 6 CFU per ml. Along with the constant increase of the colony forming units, a significant decrease can be observed in the percentage of crude oil concentration. For 3% crude oil, 70% crude oil was left over, which didn't get hydrolyzed. For 1%, it was 45%, and for C18 and phenanthrene, it turned out to be 66 and 55% respectively. Conclusion: the significant increase in numbers of coliforms parallel to the significant decrease in the concentration of remaining oil provides an experimental evidence for a major contribution of indigenous coliforms to self-cleaning of oil in sewage. This finding implies that indigenous domestic sewage microflora is capable of controlling potential spills, and for the source of hydrocarbonoclastic microorganisms, raw sewage is the most accessible and cost-effective source. The future directions of this research includes the development of novel biocompatible surfactants in order to enhance contact between bacteria and petroleum hydrocarbons, exploration of undiscovered resources of petroleum hydrocarbon degrading bacteria via new technology, and construction of genetically engineered bacteria by using synthetic biology technology so as to give more ability for petroleum hydrocarbon degradation. So that was all about my presentation based upon the chosen research article. I hope you guys enjoyed and had a great time learning something new. Thank you so much for the time.